In today's lecture, we will discuss about some important terms that are usually used in genetics. So the first term is gene. ये टर्म कॉइन की थी जोहानसन ने. एंड जीन क्या है? इट इज द यूनिट ऑफ इनहेरिटेंस. ठीक है, कहाँ पे प्रेजेंट होती हैं जीन्स? Genes are present on the chromosomes, right? And ये chromosomes हैं जो कि in the gametes they move from one generation to the next generation, from the parents to the offspring. Gametes carry करते हैं chromosomes और chromosomes पे located होती हैं genes. So genes क्या है? These are the unit of inheritance which are carried from the parents via gametes in the chromosomes to the next generations. And ये genes क्या करते हैं? इनका function क्या है? ये express करते हैं. Express करके they produce a trait. They give an expression. Where how do they express? The genes, they may interact with their alleles, they may interact with different genes, and also it may interact with the environmental factors to bring about the expression of a trait. So this is the smallest unit of inheritance that we call as gene. And now the next term that we will use very often when we talk about Mendelian genetics is alleles, or what we call as allelomorphs. Now Mendel, he said that the character, or say the trait, right, it is controlled by two contrasting genes, of which one gene is dominant and the other gene is recessive. So there are two contrasting genes for one particular character. However, in the present context, nowadays we have revised this definition, and alleles, they are defined as alternate forms of a gene. Alternate, it would be better to say if I say alternate contrasting forms of a gene. What I mean is, suppose हमारे पास gene है height of the plant. इसके दो contrasting forms हैं. One is the tall and the another contrasting character is the dwarf. या तो plant tall है या तो plant dwarf है. तो ये जो alleles हैं, say tall I represent by T and dwarf I represent by the lowercase small t. So these alternate forms of the gene height, they are called as alleles. Next term is the locus, or I take homozygous individuals, organisms, and heterozygous organisms. Okay. Suppose I have a pair of homologous chromosomes. Now what are homologous chromosomes? The same chromosomes, a copy that you inherit from the paternal parent and another copy of which you get from the maternal parent. So they are the same chromosomes, one derived from the father, the other derived from the mother. So the same chromosome called as the homologous chromosome, इस पे कोई भी एक पोजीशन, कोई भी एक साइट जिसके ऊपर जीन लोकेटेड है, उसे हम कहते हैं जीन लोकस. एंड एज सच, सिंस द क्रोमोसोम्स दे आर सेम, तो यहाँ पे भी जीन इस लोकस पे, इस पोजीशन पे जो जीन है वही इस होमोलोगस क्रोमोसोम के लोकस पे भी सेम जीन है. फॉर एग्जाम्पल, if the gene is height, so this locus, this position on the chromosome, it represents height, and same this locus also represents the gene height. In the homozygous organisms, the locus, they have identical alleles which may be dominant or they may be recessive. What I mean to say is the height is gene, the gene is height और इसका एलील क्या होगा, या तो वो टॉल होगा इफ पैटर्नल क्रोमोसोम पे इस लोकस पे इस पोजीशन पे एलील टॉल है और दूसरे क्रोमोसोम पे होमोलोगस क्रोमोसोम पे भी सेम लोकस पे अगर एलील टॉल है, right? So both the alleles, they are identical, and such an organism who has the identical alleles at one gene locus on the homologous chromosomes, they are called as homologous organism. Heterozygous in contrast क्या होगा, ये एक homologous chromosome है, right? This position represents the gene. Say again, I take the example of the gene as height. So here at this locus, when the alleles, they are non-identical, मतलब इस locus पे there is the dominant allele and इस locus पे recessive allele है. So such an organism is called as the heterozygous organism. If you see, this is also called as a hybrid. However, a homozygous organism is also called as a pure organism for that gene. Okay. Now, the next term that we talk about, another very, very important term is the phenotype and the genotype. Okay. So phenotypic means the physical appearance of a gene. What I mean by physical appearance of a gene means the expression of the gene that you can see, that is noticeable. Means what we can see with our eyes. Say, if a plant, we can easily say कि ये plant tall है, ये plant dwarf है. We can always say this flower is purple and this appears white. So just by seeing, we can appreciate two different contrasting traits of some gene. So that is the phenotype. Genotype क्या है? It is the genetic makeup of an individual, right? It is the alleles that are present on the chromosomes in an individual, which we call as the genotype, right? Say, for example, the genotype, it could be this, the gene represented by its two alleles, and this is a genotype where both of them, they are dominant and the plant appears as tall. So this is the homozygous dominant genotype. Let's say another example, capital T and small t, one dominant allele, the another recessive allele, right? So this is the genotype which is heterozygous tall, or it is hybrid. But phenotypically, if you see, both of them, they appear as tall, but genotypically they are different. This is homozygous, this is pure for the height, and this is hybrid. And both the terms phenotype and genotypes, they were coined by Johansen. Next we talk about what we mean by genome. Now genome is the sum total of the genes that are present in the haploid dose of the chromosomes of an individual, right? So that constitutes the genome. For example, in human species, we know that there are 46 chromosomes. Okay? One of the gamete would have 23 chromosomes. So in 23 chromosomes के ऊपर जितने भी genes present हैं, that constitutes the genome for the organism. Then, what is the gene pool? if I sum up all the genotypes of all the individuals in a population, so that will constitute the gene pool. बड़े simple आपको एक example देती हूँ. There is a class of around 50 students, right? तो उन 50 students, 50 individuals का जितना भी genes हैं, the total number of genes in them on all the chromosomes, if I pool them, if I take them in totality, so that constitutes the gene pool. Now we talk about two very important terms. One is known as the test cross. The other one I will discuss the back cross. Let's take test cross first. We know that when we cross two parents, right, we get F1 generation, which is a hybrid. Okay, so what is F1? This is known as filial generation, right? The first progeny, the first hybrid progeny that you get after meeting the parents, that is F1. The next would be F2 generation, which we obtain by crossing or selfing the F1 individual. So the generation obtained would be F2 and so on. So in a test cross, when we cross the F1 genotype with the recessive homozygous parent, that constitutes a test cross. What I said, I represent here by a cross. Again, I take the height as gene, gene as height, and I cross a parent that is pure tall with a parent that is pure dwarf. In the F1 generation, I get a plant, I get the seeds that are hybrid with the genotype heterozygous genotype, and when they grow, they produce again the tall plants, right? So this. So if I cross the F1 individual with the homozygous recessive parent, clear? F1, here in this case it is heterozygous tall. It could be the homozygous recessive or it could be homozygous dominant. F1 could be anything. When we cross the F1, essentially this is very important, when we cross an F1 genotype with the homozygous recessive parent, the cross is called as a test cross. Now to explain the significance of it, I make two crosses. Say my F1, I have here the homozygous tall, and in the second cross, say this is cross 1 and this is cross 2, right? In F1, I have here in one cross homozygous tall and here I have a heterozygous tall. Now if I make a test cross here, so F1 will be crossed with the recessive homozygous parent. Likewise here also this F1 would be crossed with the homozygous recessive parent. So if I make gametes, now again there are two ways of forming the gametes. See, here we get these gametes, right? And if I cross these gametes, I get these individuals. Similarly, if I form the gametes here, these are the two gametes, and here the gametes are of course the same. So to represent and cross, we can either represent it in this method. This is called as fog line method. This gamete of one parent combining with the gamete of the second parent to give you the F2 generation. Likewise, this gamete of one parent combining with the gamete of the second parent to give you the genotype. So this constitutes the F2 generation, and this way of representing a cross is called as the forked line method. Right? Another way of representing this cross is by a Punnett square, and this Punnett square, this representation was given by the scientist Punnett, and this is called as a Punnett square. Here the vertical side we represent a male, and on the horizontal side we represent the female gametes. So say this is the female gamete and this is suppose our male parent. So I represent the gametes outside the square. Now each square would represent one genotype, one individual. So capital T crossed with small t, you get the heterozygous tall, one individual. This cross with this becomes the recessive tall again, and this. So if we see, and this constitutes our F2 generation. Now if you compare the two crosses and the F2 generation that we get in both the crosses, say in cross 1, if we see F2 is all tall. The genotype is heterozygous tall, but phenotypically these plants are all tall. However, in cross 2, if we see here, we have the heterozygous tall and the homozygous dwarf in the ratio of 1:1. So with this test cross, when I cross the F1 generation with the homozygous recessive parent, I can always predict the genotype of the F1 parent, right? Supposedly I do not know the genotype. By just looking at F2 generation, we can predict the genotype of the F1 generation. So if we get all tall, right, it means that the F1 was homozygous tall. But if we get the ratio of tall to dwarf as 1:1, clearly means that the F1 was heterozygous tall. So this is the significance of the test cross. To sum up, the test cross is when the F1 individual, it is crossed with the homozygous recessive parent, and the significance is that we can determine the genotype of the F1 individual. Now, what is a back cross? Now let's take what is a back cross, right? Again, the gene is height. Suppose I cross a homozygous tall parent with a homozygous recessive parent, and in the F1 generation I get a heterozygous tall individual, right? So a back cross is one where F1 genotype is crossed to any parent, right? You can cross F1 here, this is the F1, either with the homozygous dominant parent, or I can cross the F1 with the homozygous recessive parent. So this constitutes the back cross. If we see here, I make a Punnett square, right? I represent the gamete here, right? And here I represent the gametes. So what I get is, quickly, right? So if you can see here, the ratio is heterozygous tall to dwarf. Again, the ratio becomes 1:1. But in this cross, we get, right, here we get all tall, phenotypically all tall here, and genotypically, if we say, we have homozygous tall to heterozygous tall, right, in the ratio of 1:1. So phenotypically they are all tall here, but phenotypically 50% are tall in this cross and 50% are dwarf, right? So we can say that, to sum up, back cross is when an F1 individual is crossed with any parent. It could be this parent, the homozygous dominant, or it could be heterozygous dominant, or it could be crossed with the homozygous recessive parent. If we see this carefully, this is nothing but a representation of a test cross. This I am representing nothing but a test cross. Why? Because F1 individual is crossed with a homozygous recessive parent, and this is a test cross. So we can always say that a test cross can be a back cross. Yes, a test cross can be a back cross, but we cannot say that a back cross is a test cross. We cannot say so. See, both the crosses, it concludes that a test cross can be a back cross. However, a back cross is not a test cross.